Hello everybody, good evening and welcome to your very own BYJU'S 9th and 10th grade channel. I'm your teacher Aishwarya and I welcome you to this particular class which is going to kickstart our Abhyas series where we are going to be doing the top 30 most important questions from biology, which means today we are going to be covering all the chapters in biology that starts from fundamental unit of life, tissues and improvement in food resources. We are going to be having a total of 30 questions where we will do both objective as well as subjective questions. So I hope that all of you are excited for this class. And students, very quickly before we get started, please do let me know if my audio, my video and my screen and whatever I'm writing on the screen is visible to all of you. Yes, and let me know if we are good to go. Good evening. I can see Piyush, Siri, Saloni, Astha, Sikandar, Shrivali, Preeta, Sanvi, Neha, Shrishti. I have so many students today in the class. Welcome all of you. Welcome to this beautiful class wherein we are going to kickstart your practice month. So I'm not going to waste any more time. We're going to get right to it as to what you can expect today. So as you all know, today we are going to be having a total of 30 questions. So here the first 10 questions that we will solve are going to be your objective questions, which means that we will have 10 objective questions and I will be giving you YouTube polls for the same. While on the other hand, when we look at the next 20 questions, we will be doing subjective. Now here, out of 20 subjective questions, we are going to be doing 10 2-marker. So they're going to be easy ones, right? While on the other hand, we will be doing 10 which are going to be case-based and long answer format questions. So here, this is how we are going to be doing it today. So very quickly, students, can you tell me if my video quality, if my audio, everything is fine? All good, na? No issues? So I can see that it is all good to go in my screen right here as well. I am seeing that we're good to go. Okay. Ayushman, you're going to rock your exam. I know it. You have my blessings at all times. You're going to ace it. Okay, all good. Ma'am, video quality isn't so good. So for those of you who are experiencing poor video quality, please go to settings, right? Then go to advanced settings, okay? And in advanced settings, you will have an option where you can set it to 720p. So please kindly do this for those of you who are experiencing poor video quality. Yes? I understand some of you are in parts of India which is experiencing cyclone. I hope all of you are safe. All of you are, your families and you are all safe and healthy. Please take care of yourselves. Anish, you also have my blessings. You have all of our blessings. Settings ka option hai, in the side it is available. Yes? Now, when you come to class, what would I expect you all to do? Small things, right? Small request that you have your notebooks and textbooks. Have your stationery ready, have your water bottle ready and a quiet place to study, right? Now, if you are going to spend around 1 hour, 30 minutes with me, that means that you are dedicating that time to practice. So every day in your timetable, you decide that you want to practice questions, right? So what will you do? You will dedicate that time and don't get distracted. Don't have parallel conversations because this way 3 chapters will be revised effectively, which is why I request all of you that when you're in class right now, please pay attention. Don't get distracted. 1 hour, 30 minutes. Yes? Okay. Afia, if that is the case, I request you to let us know in the comments of this video, right? Now, a quick reminder, see, for Abhyas series especially, every week we will put up a timetable. So, half the time you guys keep telling, "Arey ma'am, we are not very clear of when what classes happen," which is why we will give you a weekly timetable so that you can plan out your entire week and you know what you can expect from us. So as you know, the first class in Abhyas is going to be from biology where we do top 30 questions. Tomorrow you have 2 classes. So tomorrow is a very long day for you because you have 20 questions with Harsh ma'am, along with which you will have a quiz with Kriti ma'am. But after that you have a break. So 6th you don't have any class, but again on 7th and 8th you have classes again. So as you all know, this is going to be your timetable. Those of you who have not seen the community post, those of you who have not seen the timetable, this is once again a reminder for all of you to update the timetable and take a screenshot and keep it ready. And every week for Abhyas, we will be updating your timetable. I know a lot of you are saying, right? A lot of you here are saying, ma'am, why class 10th is getting attention? They have their pre-board exams and boards coming up, but we are not going to reduce the focus on you 9th graders at any point, right? So don't worry. You can do that, Ayushman, you can do that. That is up to date with competency, so you can watch that particular session as well. Okay, is Menti for chemistry? Yes, Menti is for chemistry. Okay, so now that I've given you a quick download of what you can expect, jaldi se we will get started. Yes? Shall we get started? Awesome. First set of questions is going to be objective, right? Physics would be from Saurabh sir. Yes, physics would be from Saurabh sir. Today, no Menti. Okay? So let's get started with question number 1. YouTube polls on your screen now. Which among the following is not a function of the vacuole in plants? They are responsible for storing some metabolic waste. They help with the process of cell division. They help in maintaining turgidity, or they provide structural support. The poll is live on your screens and you can give me the answer here. We are talking about the function of which is not a function. So if you have to tell me which is not a function, then what should you know? You should know what are the functions of vacuole. Now in the case of plant cells, what do we know? Plant cells have large vacuoles, right? And what do? They help in, storing excess water, and some amount of waste material also gets stored. And along with this, because they store water, they also help in providing shape to the cell, right? So we see that they give structural support and they maintain turgidity. But do they help in cell division? Are they responsible for cell division where there is increase in number growth? No, vacuoles have nothing to do with it directly. Which is why, what do you see? Like how many of you have told me, ma'am, we can lock option B. Exactly. We can lock option B, and the correct answer here is going to be option B. So well done all of you. I'm going to end the polls and I can see that majority of you have given me the correct answer. So that is why in this particular case, the correct answer is option B. They are not responsible for helping you with cell division. So in this case, for objective, all of you have scored 1 out of 10. So well, very good. Ma'am, can you tell me about the biology quiz? That will happen next week most likely. Okay? Ma'am, do they get divided in cell division? See, during cell division processes, there are certain cell organelles that duplicate or that may form later on as well, right? So in this particular case, their contribution in cell division, they don't drive cell division as a process. But yes, when the cell is formed later or when the daughter cells are formed, vacuoles are also formed, okay? Awesome. Let's go on to question number 2. First, I'll read out the question and then give you the polls. Meristematic tissue in plants are what? They are localized and permanent. They are not limited to certain regions. They are localized and dividing cells, or they grow in volume. Yes? So you have to tell me which is correct about meristematic tissues. Somebody asked me, ma'am, is it going to be the nucleus which is involved in cell division? Yes, nucleus has a very important role. And students, I forgot a very important thing. Today, by the end of this class, I have a surprise for all of you, right? I have a surprise where I will give you some bonus questions for practice, and that will be as a form of a test. So everybody stay with me till the very end, because you will have to, give that test. And all of you will get a chance to get featured on our community page as well if you give the correct answers within the first hour. So I'll give you more details of it towards the end of the class, but stay with me till the very end. Yes, I was giving you some updates, so I forgot to give you the poll, but the poll is now live. So we're talking about meristematic tissues, right? So as you all know, we know that plant tissues can be broadly classified into 2. We have meristematic tissues and we have permanent tissues, right? Now, meristematic tissues are those tissues which are constantly dividing. Yes, so they are constantly dividing and they are increasing in number. Which means, where do I find my meristematic tissues? I will find it in the growing regions of the plant, right? Root tip, shoot tip, I will find them in all of these regions. But if you see, Mainly per se meristematic tissues, although they are found in such regions, they don't have a specific shape, size, right, or a specific function to do. Their function is to just keep dividing and dividing and dividing, right? So they are relatively unspecialized. So I cannot use the word permanent, because anywhere where the term permanent comes into the picture, it means that they have a specific shape, size and function. So no. Now it says they are not limited to certain regions. That's not the case. They are limited to certain regions, right? We know that meristematic tissues are limited. Where are they limited? They are limited to the growing regions of the plant. So no, this statement is also incorrect. Do they grow in volume or do they grow in number? Now, meristematic tissues, as you all know, right, they are responsible for growing in volume or they are responsible for growing in number. What can you tell me? It is, of course, dividing in number, right? Which is why in this case, what do you see? They are localized. Localized means they are found in specific regions. Yes? And what are they? They are actively dividing cells. So the correct answer, as all of you have told me, is nothing but option C, which is correct. So very good. All of you have now got 2 out of 10 here. Very, very good. Now, in the meanwhile, students, if you are enjoying the class, don't forget to hit the like button on this video. We should have minimum 100 likes, right? Minimum. So, fatafat, go and hit the like button as well. Now, let's have a look at the next question, right? Next question on your screen. Here we have a cell organelle, and here if you see, there are 2 parts given, X and Y. Now, focus on where the X is being marked. X is pointing towards some finger-like projections, while Y is pointing not, Y ka I will change it a little. Y is pointing towards this membrane here, right? The labeling is incorrect, so ignore this. This is Y, right? Y is pointing towards the membrane. So can you tell me which organelle this is? Jaldi say. We see that it is a double membraned organelle. It has some finger-like projections. Awesome. So which organelle? Exactly. So this right here is going to be mitochondria. And like I said, Y, so X right here is with reference to the finger-like projections. So what are these finger-like projections? Can you tell me? The finger-like projections in mitochondria are called as, very good Tanya, it's called as cristae. And here Y is pointing towards the membrane. So here it is pointing towards the inner membrane, right? So that's a small incorrect labeling that is there. Now, not the matrix. Matrix nahi, it's pointing towards the membrane, right? Okay. So based on this, this is the question now. Based on this, can you tell me which is correct with respect to X and Y? See, here, okay, if you take X as a labeling of matrix also, look at it as the membrane, right? Think of it as the membrane that is there. We are talking about Y as the membrane. So based on this, which is correct? X increases surface area for ATP synthesis, Y increases surface area. We see that X is responsible for increasing surface area for proteins, or Y increases surface area for protein synthesis. So which among this would be true? Yes? Ma'am, there will be 2 correct answers. No, only 1 correct answer is here. That's why I said Y ka labeling, because what happened was we highlighted it, okay? You can't see the actual markings on the image. So we added another line on top of that, and when that line got added, there is an incorrect, there's a slight mistake where it has extended beyond. That's why you think it's matrix, but it's not matrix. Ma'am, what is synthesis? Sagar, synthesis means to produce something. So if I say synthesis of ATP, it means to produce ATP, right? So that is what we mean by synthesis. So based on this, what would be the answer? Very good. I can see the answers coming in, and majority of you are giving me the correct answer. Bacha, synthesis is not breakdown, Hema. Breakdown is not, see, when you say photosynthesis, that means you are synthesizing food in the presence, right? It cannot be breakdown. So breakdown is incorrect, okay? Very good. All of you have given me the correct answer. Majority of you have given me the correct answer, and it is option A. See, what is mitochondria? Now, mitochondria that is there is basically, It is a double membraned cell organelle, right? So it is a double membrane cell organelle. Yes, that means it has an outer membrane and an inner membrane. Now, what is the main role? Often mitochondria is called as the powerhouse of the cell, right? So we call it as the powerhouse of the cell. Why? Because we see that this particular cell organelle is what is responsible for producing energy. Now, how is energy produced? Energy is produced in the form of ATP. Now, where exactly is this ATP produced? You see, the inner membrane that is there is made into finger-like projections. So, if you see, this was Y, assuming this was Y, which was marked as the membrane. I'll not say inner membrane, we'll just call it as membrane. And this was X, right? So, X right here is pointing towards these finger-like structures. So, the inner membrane of the mitochondria is folded into finger-like projections called as cristae. Now, why are there finger-like projections? Because we see that these finger-like projections increase surface area for the production of ATP. That means that more and more of ATP is produced because they have increased the surface area. Now, we also see that mitochondria is an example of a cell organelle that has its own DNA, right? So, it has its own DNA. That means it synthesizes its own proteins, which happens in the matrix, right? But here there are no finger-like projections that increase surface area for protein production. Yes? So which is why from this case, what do we see? Answer is option A. Now you're getting confused, right? You are getting confused wherein we see that this Y ka labeling went wrong. But Y was pointing not towards the outer membrane, right? I mean inner membrane, but Y was pointing towards the outer membrane. Yes? Gurlal, they're still working. By Thursday it should be done, so that it will be available on a website, so that directly website say you can download. So what they've told us is that they've got back and they said by Thursday it should be done. Yes? Okay. So what is ATP? ATP stands for adenosine triphosphate. So this is the answer. Are we clear? Yes. It's called, for this reason, yes Tripti, it is called as a semi-autonomous cell organelle. Yes? It is going to be 2 hours today for sure. Tissues, you can ask me, Raji, when we have a question. Now I'll end the poll so that all of you, we can move on to the next one. Ma'am, through this, will bio revision be done? Animesh, Atharva, to an extent you can do this. But higher order questions, we may not solve many of it. You can always watch the Anthe sessions that we have done because they have higher order questions also. Yes? So all of you have got 3 out of 10. Now moving on to question number 4. While observing human cheek cell under a microscope, students will not observe what? Nucleolus, mitochondria, nucleus, chloroplast. This is easy peasy, lemon squeezy question. I don't even think you need poll for this, but I will still give you. Otherwise, you all will accuse me telling, ma'am, aapne to poll nahi diya. You told me poll, so I will give you poll. But by the time I type and finish, you guys will give me the answer. Now quickly, human cheek cell, right? Human cheek cell is your keyword here. So first and foremost, when you see human cheek cell, is it a plant cell or an animal cell? What is a human cheek cell? Is it plant or animal? Now in this case, it is very easy, right? We know that human cheek cells come under the category of animal cells. And what do animal cells lack when compared to, let's say, your plant cells? They don't have cell wall. They do not have plastids, right? So in this particular case, what is not observed or what will they not see? They will not see chloroplast, because chloroplast is a plastid which exclusively is seen in plant cells, right? And chloroplast plays a very important role for the process of photosynthesis. If we had chloroplast, we will also be making our own food. Yes? Which is why here we see that plastid is something that we exclusively see. Siri, vacuoles also. I would correct that statement and say large vacuoles are not seen in animal cells. They may be very small or they may be absent, right? So vacuoles, large vacuoles are seen in plant cells for storage. Okay, improvement in food resources, I'll come to that, Abhimanyu. I will give you some strategies when I maybe take up some questions if you're okay with it. Okay, so as you can all see, you have all got 4 out of 10. Very, very good. You guys are acing this quiz. Very proud. Now let's go on to question number 5, which is assertion reason. Assertion statement says that most plant tissues are dead. Reason being that due to sedentary existence, that means they are found fixed at one point. Plants require dead cells that provide mechanical strength, they have more amount of dead cells that give them mechanical strength, let's say, when compared to living cells that need more maintenance, while your dead cells require less maintenance. So in this particular case, what would be the answer? Okay, so let me quickly give you all the polls. Now, for students who struggle a little bit with assertion reason questions, right? Because I know assertion reason questions can be tricky. Now, for many of you who've been regular to my classes, you know the technique, because I have only one-stop solution technique for all of it. But if it's the first class you are attending with me or watching it, right? See, you have assertion reason. Take it as two statements. So basically, this whole process requires you to do two steps, right? Step number one is to identify them as individual statement. Look at it as statement 1 and look at it as statement 2. Now what you need to do? Identify if they are true or false. Now if any one is false, no, that first two statements will the first two options will get eliminated, which says both A and R are true. If any one turns false, you can immediately identify that. But then if both statements are true, you will go into step number 2 to identify if reason is correct explanation. Now, how will you know if the reason is correct explanation? Easy. After the assertion statement, put a full stop and ask yourself why. And if this reason justifies the assertion statement, then only it is a correct explanation. If it does not answer questions of why, how, what, where, then we know that this right here is incorrect. Yes? So in this particular case, most of plant tissues are dead. That means that when compared to a human being or an animal, right, the percentage of dead cells that we find, the percentage of dead cells that we find are more, right? So we find more number of dead cells in plants. So yes, this statement is true. Now, why is it that we find a greater percentage of dead cells? Because plants don't have the ability to move around and protect themselves, right? Plants are found fixed to the ground. So they require more amount of mechanical strength, rigidity, protection required, because they need less maintenance. Unlike your living cells, because when a cell is living, it requires constant supply of food. We know that it needs energy. So we see that the maintenance is more. Which is why if you see, this is also true and this justifies the answer. Which is why the correct answer here, as many of you have given me, is option A. So are we clear with the answer? Are we clear on how we have arrived? Yes? I am doing good, Sumit. Badhiya, sab badhiya. How are you? Ma'am, ye samajh mein nahi aaya hai. See, Z, think of it as, see, do you see plants moving around? Nahi na. Plants are all found fixed to the ground. We don't see plants going and getting water, eating food outside. No. They are found fixed to the ground. Which means that if there is, let's say, climatic conditions that we see, right? For example, there is a cyclone as we experience right now. Or if, let's take the example of cyclone itself. We are all able to protect ourselves. We are able to protect ourselves, let's say in our houses. We are able to move around and we are able to do that. So in our case, we are able to physically protect. But in the case of plants, they cannot run away. No, cyclone is coming, now I have to run. No, they are found fixed. So within their body, they have more percentage of dead cells that acts as a barrier, right? That gives them rigidity and provides protection. So yes, they do not exhibit locomotion. That is why they need more cells to give them protection. That is the reason. Why ATP is the energy currency? ATP is energy currency simply because we see that the energy stored, right, for any chemical reaction, in the expense of ATP, we see that we use energy. Chemical energy is used, right? Okay. Now let's move on to question number 6. 5 out of 10. I'll pick up the pace a little bit. Abhi thoda we are going slow. So I'll pick up the pace now. Okay. Awesome. The tissue that is thin enough to allow diffusion. So basically, which animal tissue allows diffusion? Cuboidal epithelium, squamous epithelium, areolar connective, or is it cartilage? This is very, very easy. You need to identify the function. I don't think you need the poll, but I will still give it jaldi jaldi say. right? Now we're talking about animal tissues which allows for diffusion. Now where does this process of diffusion happen? This process of diffusion that is there happens in the alveoli, right? So alveoli that are there are these balloon-like structures. Yes, balloon-like structures which are normally found in the lungs. Yes, and if you see the inner lining of the alveoli are made up of flattened cells which have a prominent nucleus that allows gaseous exchange. That means gases can go from a region of higher concentration to a region of lower concentration, right? So we see that this movement of substances from region of higher to lower concentration is what we call as diffusion. And where do we see, or what has it? Simple squamous epithelium, right? That inner lining of alveoli is mainly made up of simple squamous. Skin pores. See, pores on our skin are mainly for release of sebum, that is oil, or for release of sweat. But other than that, we cannot do gaseous exchange with our surface, right? So the upper surface of our skin is mainly made up of dead cells that are keratinized basically. That again mainly provides protection. Yes? Okay. So very good. Very, very good. All of you are giving me correct answers. 6 out of 10. Now moving on to question number 7. You have 4 statements given from which you need to tell me which is the incorrect or wrong statement, right? So can you quickly tell me? Now I'm going to give you the polls in the meanwhile as you all go through it. And students, if you are enjoying the class, don't forget to hit the like button on this video. Very, very important that you like this video. Okay, single membrane cell organelles that are there include your ER, right? So basically, unlike your mitochondria, chloroplasts, which have inner and outer membrane, they will have only one single membrane. So they are called as single membrane organelles. Yes? What are the functional parts of the cell? Saraswati, functional parts of the cell include all your cell organelles, right? They carry out specific functions. So they are functional parts. Why do herbivores have larger small intestine when compared to carnivores? I think you are a 10th standard student, but I will still give you the answer. Larger small intestine, because if you see the kind of diet that it comprises basically because of its diet, they take a longer time to process their food. Carnivores eat meat, right? So they need to process the food quickly. They cannot store too much of it in their body. Hence, that is the case. Very good. Today I feel like I'm on a roll, right? I feel like I'm a rapper and I'm going with all the answers and I'm going with the explanation very quickly. Okay, let's have a look one by one. The nature of matrix differs according to the function of the tissue. Now, if you look at the concept of matrix, right? Concept of matrix comes in when we look at connective tissue. Yes? Now, connective tissues are those tissues which are extensively found in our body that connects and binds different parts of our body together. Now, we know that we have loose connective tissue, we have dense connective tissue, and we have fluid connective tissue. Now, what is different? We see that what is different is the matrix. And the nature of matrix depends upon the kind of function that they carry out. So, yes, this statement is true. Now, fats are normally stored below the skin and between internal organs. Yes, we know that normally if fat is found beneath the skin, right, we call this as subcutaneous fat, right? And why do we have this? Because fats that are there or adipose tissue that is there is responsible for giving insulation, right? For, sort of protecting, for maintaining the body temperature. And if you see the internal organs, some of the internal organs are surrounded with fat. We call that as visceral fat, right? So that is also true. Now we see that cells of striated muscles. What are striated muscles? They are our skeletal muscles, right? Now if you remember skeletal muscles ka picture, we see that skeletal muscles are long, right? We see that they are striated. They have alternating dark and light bands. We see that they are multinucleated. They have many nuclei and they are unbranched. Very, very important. Because if it becomes branched, it is cardiac. So here, this is also true. But epithelial tissues, right? Epithelial tissues have intercellular spaces. Do interepithelial tissues have spaces between them? Yes or no? Do epithelial tissues have spaces between them? No. We don't have so in epithelial tissues, I always tell you it is a continuous lining. That means it is continuous. There are no spaces present. And what is intercellular? Between two cells, if I find space, I will call that as intercellular. Inter means between two. So in this particular case, which is wrong? It is option C, that epithelial tissues have intercellular spaces. Ma'am, cardiac muscles are less striated and uninucleated. Then how? See, cardiac muscles, on the other hand, are branched, uninucleated, and they are structurally different. See, they may have an appearance which is similar to skeletal muscles, but they are not the same. Because of their striations, they may appear similar, but there they are shorter, they have intercalated discs, they have other structures. So that is why they are different. Okay. All the best, Vandana, for your exams. I hope you do well for that. Which book do we use? We use all possible resources, right? Whatever books are available in the market, we go through all of it. We have an internal repository, and that is where we get the questions from. Okay. Ma'am, आपको ठंड नहीं लग रही है? बहुत ठंड लग रही है, but Ankita ma'am has my sweater. Okay. Yes. Ma'am, growth of pollen tube after fertilization. Moon Bacha, you tell me your name. I think you are in 10th standard, no? I can see 10th standard students coming and asking me how can I learn dihybrid cross. Students who are in 10th, please, if you don't mind, come towards the end of the class so that I will give you strategies. But now I'm teaching the 9th graders, so it will be difficult for me to sort of take your doubts, okay? Full form of ATP, adenosine triphosphate. ठीक है. Jaldi say students, don't forget to hit the like button on this video, and you know that I have a surprise for you towards the end, so stay with me. Come towards 6:45, I'll take your doubts then. Ma'am, muscular tissue and its type. Muscular tissue, I have a question based on it, so I will be able to give you the answer. Okay. Ma'am, growth of pollen tube, nahi. Ma'am, that is not fertilization wala question is 10th ka. 9th mein you don't have that much. Okay. Arey, math ka question you ask Harsh ma'am tomorrow. Yes? Next up, question number 8. To enhance fish seed quality, now worked on to enhance fish seed quality that is now being worked on, like basically to improve the quality of fishes in ponds. How do we do that, right? How do we do that? Do we do hormonal modification? Do we do gene transfer? Do we do hormonal stimulation? Or do we do gene abbreviation? I will give you the polls. मैम 10th के क्वेश्चंस पूछिए. 10th का सेपरेट क्लास होगा बायो का नेक्स्ट वीक. यस, then we will do this week. We are going to be solving sample paper. I like how they've come and said 10th is very hard. But when BYJU'S is there, you have nothing to worry about, no? You have us. Awesome. So basically, we're talking about in culture fisheries, right? Or when we do culture fish farming, we know that we are culturing the fish, right? So we are culturing the fish. So in order to increase the fish seed quality, or basically the fish seeds are basically the eggs that are there, right? So in such cases, in order to increase the quality and number of it, what can we do, right? What can we do? Can we do hormonal modification, right? Should I modify the hormones? Yes. Do we have to do gene transfer, or do I have to do gene abbreviation? Or simply put, can I stimulate it with hormones where more number of these fish seeds or eggs are produced? Yes? Okay. Chitin sugar, Prisha, is basically the sugars or the substance that makes up the cell wall in fungi. Kartikeya, I have a question on this. Differentiate between hypo, hyper, iso. So if you don't mind, but it's towards the end, okay? I think question number 26 or 7 is this question. If you don't mind waiting till the end, will you please stay with me? I'll explain that. Muscular tissue also, question number 24 or 3 is that question. So jaldi se I will do that. Yes? Stay with me till the end. I'm going to solve all your doubts. Now, yeah, somebody was asking me what will happen if RBC is kept in hypertonic. Hyper may it will shrink, right? Okay. With this, I can see many of you have given me the answer. Tarun, they are helping you out, right? What is function of ATP? Full form of ATP, Sagar, I told you. Adenosine triphosphate. Function, energy currency. So yes, it is responsible for hormonal stimulation, or by doing hormonal stimulation, I am able to improve fish seed quality. So here all of you have got a perfect strike. Yes, you have got the perfect strike of 8 out of 10. 2 more questions to go. Now let's have another assertion reason question on your screen. See the doubts that you guys are asking me, I have questions based on that, students. So please stay with me. For squamous epithelium, all the functions and everything, I have questions based on it. So stay with me, I will clarify all of it. Yes, we'll pick up the pace also. Assertion statement says that green gram is a kharif crop. Reason being that green gram is completely grown in month of June to October. Tripti, I will answer your question. Sagar, I have explained it to you. If you want me to explain it once again, how about we finish this whole bit and then go? Because I am taking your doubts. Gurlal, Abhi, I will send it to you on Telegram because the PDFs are still being uploaded onto our website. But till once the website is functional, then you will be able to get it directly. Poll is live, bacha, YouTube pe. Students, help out Sagar because I think he's missing out my explanation. So please help Sagar out in the chat as well. Yes, Pragati, we'll be fast. See, in this particular case, is green gram a kharif crop? Now, many of us are not clear whether green gram is kharif or no. Is green gram kharif? Yes. Is green gram kharif? Yes or no? See, pulses, I know, are little tricky. We know soybean, okay. Soybean, fine. Jaldi say, is green gram kharif or is it rabi? No. See, pulses in general are tricky to learn, right? Pulses are little tricky to learn because pulses is a broad category. Now, if you look at pea plant and all, pea that is there is a type of leguminous plant, but it is rabi. But here we're talking about green gram. Green gram is kharif, right? Yes. So, green gram here is kharif, which means this is true. Now, when are kharif crops grown? They are sown during June, July month, right? Or July, August, but mainly in the monsoon season, right? But here during October month, it is harvested. So, in this particular case, both are true. So, green gram is a kharif crop? Yes. Why? Because they are sown or they are completely harvested between the months of June to October. So, in this particular case, what will be the correct answer? The correct answer here is going to be option A. Yes. So, answer here is option A. So, here all of you have got, I can see many of you, I would say, is given me the correct answer. Very good, students. Very, very good. Ma'am, ये 10th का question नहीं है. No, this is 9th का. You have kharif, rabi, zaid crops in improvement in food resources. So please pay attention. Hema, if it is not part of ICSE and you are ICSE, you can try answering. But it's okay if you make some mistakes. Okay? Awesome. Now let's have a look at the next question. You have an image given to us where a bacterial cell is given, where 1, 2, 3 is given. Now, there are 4 options from which you need to tell me which is a correct combination of 1, 2, 3. Now, before I give you the options, can you tell me, can you label from the image and tell me what is 1, 2, 3? Yes? This season we will do menti, aaj to menti nahi hai. Okay? What is the outermost? Oh, Shaurya, okay, I will keep that in mind. See, 1 is pointing towards the outermost covering. What is the outermost covering? Yes, outermost covering that is there is going to be the cell wall, right? Outermost covering is cell wall. Now, option 2 is pointing towards this, right? It's pointing towards this. Now, this could be one in 2 things. It could be genetic material, right? Or we know that the region where the genetic material is found concentrated will be nucleoid. So, it could be one in 2 things. Then what is 3? Very good, Tanmay. Very good, Zaid. Yes, very, very good. Naitik, chromosome nahi hai. Genetic material abhi ke liye rakh lo. Very good, Kriti. Deepak, awesome. Awesome, Kartikeya. 3 is going to be cell membrane. Now, if you've identified these, can you tell me which is the correct option from the following? So, let me give you the poll. Now that you've identified what is A, B, C, I mean 1, 2, 3, you can give me the answer on looking. So, the way I'm going to solve, right? The way I solve these questions should be the methodology that you can try upload. I mean, you can try using, wherein you get an idea of, okay, if I have a diagram based question, the first thing to do is not to look at the options, but the first thing is I will neutrally have a look at the diagram where I will solve it. Once I solve it, then I will go ahead. Yes? Okay. Afia, I will take that question, very important one, right? So in this particular question, if you see, what is nucleus? Now you know that cells can be broadly classified into two. We have eukaryotic cells and we have prokaryotic cells, right? Now what are prokaryotic cells? Prokaryotic cells do not have a well-defined nucleus. That means they have no such thing called as nucleus. So if you see, they'll see that the genetic material is found concentrated in one region of the cell. And this region where the genetic material is found concentrated, we call this region as nucleoid. So where will I find my nucleoid region? Only in prokaryotic cells. Yes? While in eukaryotic cells, we see that there is a true nucleus, right? Or a proper well-defined nucleus. So if you take the cell, there will be a double membrane nucleus. Then we see that there will be a dark concentrated region. So this whole structure is called as nucleus, right? Then this dark concentrated region you see is what we call as nucleolus. And this nucleolus has a very important role to play when it comes to production of your ribosomes, right? Okay. Ganga, I think I might not be there in that class, but Ankita ma'am will be there. Thumbnail is not updated. So in this case, yes, all of you have given me the correct answer. Awesome, all of you. Very, very good. So in this particular case, what do we see? All of you have got 10 out of 10 for objective. Yes. So this is what we mean by basically you got 10 out of 10 in your objective questions. Now we will move on to subjective questions. Okay. Are we ready? पहली बार class ले रही हूँ. I'm so happy to hear that. ठीक है? Thank you so much. See, angiosperm category and all of that comes under diversity chapter, which is deleted from CBSE. But in case I will do a bridge class for all of you, maybe during your winter vacations, so that even though it's not part of syllabus, you will still be prepared for your 11th and 12th. So for my students who wish to pursue NEET, biology in 11th and 12th, this is for you. Yes? No problem, Asta. Always come back. See, simple squamous epithelium, for those of you who have doubts, I have a question coming up very soon. Please stay with me. Stay with me because I will take all your doubts. Don't feel like, ma'am, to doubts nahi solve karegi. No, I will take your doubts, Harleen, Tripti, don't worry about it. Okay, so we'll have a look. Francis, I know that is slightly inconvenient, but I hope you are enjoying our classes on YouTube. If you are, let me know that also, right? We should also spread good things, at least positive things. ठीक है. Ma'am, आपने natural resources कराया है. See, Pragati, that's part of your assignments. So we've already done it earlier on the channel. So older videos are available, but for current syllabus, though, we will not be doing it. Okay? Yes, Abhimanyu, I will do diversity. Pakka. I'll take a long class and I will do it. Question number 11, students, I need to pick up the pace. Yes? Okay. While watching Discovery Channel, RF saw that whales, polar bears, seals all have thick layers of subcutaneous fat in order to survive tough regions. Explain how survival of such animals is related to subcutaneous fat. You have 2 marks to give me the answer. Can you tell me? What is the answer? There are 2 keywords that you need to give me here. Exactly. So subcutaneous fat that is there, right? Especially in animals like your polar bears or animals that we find in, extreme cold conditions. What do we see? They have a thick layer. And what do we call this thick layer as? We call this particular thick layer as blubber. Now, what is the function of this blubber? Now, we know that adipose tissue that is there not only helps in storage of fat but also acts as an insulator, right? So they help in conserving the body heat, and thereby we see that in such extreme conditions they are able to survive. So in this case, what do you observe? We see that the fat depositing layers found underneath the skin, right? Underneath the skin organs is what we call as the subcutaneous layer of fat of adipose tissue. They act as insulators and prevent heat loss. Now in whales, polar bears, they have a thick layer so that they can maintain their body temperature. So you need to give me these 2 pointers, right? These 2 pointers if you give me, I will give you 2 marks for this particular question. So very good all of you, very, very good. Subhashri, areola tissue nahi hai, adipose tissue. Yes, adipose tissue. Charan, I know cyclone is there in your region. Please be safe. Please take care of yourself. My prayers and wishes. See, we know about adipose tissue, right? What is adipose tissue? It is a type of loose connective tissue, and they are made up of specialized cells called as adipocytes. Now, what is the function of adipocytes? They have the ability to store fat, right? So their function is to store fat. Now, adipose tissue's function is not just to store fat, but we see that it can act as an insulator also. What is an insulator? It means that it will not allow heat to escape from the body. Thereby, it will store or it will maintain the heat. Now, there are 2 kinds based on where we find the fat, right? Fat can be found beneath the skin. Yes? So, a layer can be found beneath the skin. If it is found beneath the skin, we call it as subcutaneous. Beneath the skin means subcutaneous fat. But if it is found in and around our internal organs, we will call it as visceral fat. What is visceral fat? In and around our internal organs. Yes? Okay. Ma'am, definition of fat. See, fat tissues are basically adipose tissues that store fat, right? Awesome. Ma'am, are we cold-blooded? No, we are warm-blooded animals, which means that we don't change our body temperature with respect to our surroundings, but we have a fixed constant body temperature that needs to be regulated. Cool. Now that we are clear with this, we will go on to the next question. Yes? Okay. Why should preventive measures and biological control methods be preferred for protecting crops? Now, this is a 2-mark question. And this is with respect to why we should do crop protection management. Why should I do crop protection management? And what am I protecting my crops from? Yes? Thank you, Francis. Thank you so much. Yes, carbohydrates gives a large amount of energy, correct Amrita? But it gives us instant energy, no? But our body needs stored energy also. When we don't have carbohydrates, it's not like our body is like, oh, carbohydrate is not there, I don't want energy. We need to store it as well. So that way we see that it is in the form of fat. Yes? Ma'am, to get good crop. So that pests and to protect us from pests and fungi. Ye chapter nahi hai syllabus mein. If you're CBSE student, you will have it. If you're some other state board, it will be difficult. Very good. Exactly. So why do we have preventive measures or why do we have biological control measures? Because if you look at it, crops are normally attacked by pests. They are attacked by insects. Sometimes even weeds hinder their growth, right? Which are unwanted plants. So preventive measures are normally taken. Like what? Making sure that, we prepare the seed bed properly. So preparation of seed bed. So whenever we sow the seeds, we make it a point that there are no unwanted seeds there, right? So making seed bed preparation, making sure we sow the crops properly at a timely manner, ensuring that we practice methods like intercropping. All this will help in getting rid or tackling weed removal, right? Or preventing unwanted growth. Similarly, to tackle pests and insects, we can grow resistant varieties, right? Crops which are like, your BT brinjal and everything which is pest resistant, we can grow such ones. So why are we doing this, right? What is the reason for having preventive and biological methods? So that end may we are able to grow crops that are safe and we get high yield, right? So we take preventive measures like the ones I stated to get high yield, and that is ecologically friendly or eco-friendly such that it does not harm, right? But at the same time, if we use way too many chemicals, we use too many pesticides and insecticides, weedicides, that in turn can have an impact. Which is why here these are the two steps that you need to write for why preventive measures and control measures are necessary. If you write this, 2 marks. If you support this with an example, right? If you support it with an example like some of the methods I told you, using, doing the preventive measures I listed, if you are able to give me in detail, I'll give you marks for that as well. Ma'am, what is yield? Yield is basically how many crops you harvest per area, right? So more the number of healthy crops you harvest, more the yield. Yes? Okay. So Soni Singh, I hope you are clear. ठीक है. Moving on to the next one. This is a question that we have already discussed, right? Mention desirable agronomical characteristics for crop improvement, right? Desirable agronomic characteristics. Okay, you remember this is with respect to crop improvement strategies, right? So with respect to crop improvement strategies, we know that why we do this. Yes? Nikar bacha, what did you not understand? Yeah, tallness, dwarfness, exactly. Very good, very, very good, right? Can you tell me what you want to repeat? If you tell me generally, it will become difficult. So here specifically, when you talk about crop variety improvement, we know that there are various objectives, right? And one may be have desirable agronomic characteristics. Now, what are desirable agronomic characteristics? There are 2. One, we see that tallness and profuse branching is preferred, right? So profuse branching is preferred for fodder crops, right? This is one desirable one. Similarly, we also see that dwarfness, right? Dwarfness is a characteristic we prefer in cereals. So in order to achieve this, we see that we aim for improving the variety. Yes? So what is tallness and dwarfness? Tallness is basically the height, right? Height of the crop. So for fodder crops, we see that what is fodder basically? Fodder is basically what you give to the animals. So for those, we prefer to have taller crops, right? We prefer to have taller crops which have branching in them, so that even if you have, so the number wise, if you produce less also, we see that we are able to give them as fodder because they have that height. While on the other hand, what do you say, for cereal crops like rice, wheat and all of that, dwarfness, height is not preferred, but for it to be relatively dwarfer is something we prefer. So that is what we call as desirable agronomic characteristics. What are some desirable features I would like to look? Yes? So in a nutshell, if I have to explain crop variety, nigger and many others, what is crop variety? See, a crop that is there, any crop, rice, wheat or maize if you take, if for it to be almost, in my eyes, if I want to make it perfect, right? If I want to make it perfect, I will want to have a crop which is growing, giving me lots of seeds, which is giving me the perfect kind of crop that I want to grow, right? Imagine in all ways I want it to be perfect. So that way, if I look at it, there is no crop which is perfect, but I can constantly improve the existing crop. So crop variety improvement aims at improving the quality or the variety that exists. Maybe ones that gives me more yield, more number of crops, that has improved quality. For example, if I want to grow wheat and the purpose of me growing that wheat is for baking, then I would want the seeds to be a certain way. So that way I'm able to improve the variety as per my need. Similarly, it should be able to grow faster. It needs to have wider adaptability. Yes, so in this particular case, crop variety looks at all of this. So in this case, as you see, right, profuse branching. So these are all desirable characteristics for crop improvement, but they are asking you for agronomic traits. So this I would say is something we can ignore, and you can write the answer as tallness and profuse branching. For what? In fodder crops. and dwarfness in cereal crops. So this would be the answer. Dwarfness means height thoda kam hai. That's all. ठीक है? After question number 15, I will give you a water break. After question number 15, water break. Yes? Okay. Agronomic means agricultural, right? Agricultural characteristics. Is it so, Charan? Please take care. Please take care. Stay at home only. Okay. Ma'am, what is profuse branching? That means it branches a lot. That's all. Simple. बहुत सारा branching होगा. Next up. What are the characteristics of xylem? Okay. Why are xylem and phloem called as complex tissue? Easy peasy. No problem. Okay. Yes. Simple question. Why is xylem called as a complex permanent tissue? Why are you scaring the 9th graders for 10th? 10th is easy only. 10th is not too hard. Or jaldi say students, I can see that many of you are not liking this video. Very sad, huh? All of you are saying, Ashwara ma'am, you're helping us, but you don't want to show me your love. How will you show me your love? By hitting that like button. Do not forgetting to hit the subscribe button. These two things are important, right? Now we know that both xylem and phloem come under the category of your complex permanent tissue. Now why are they called as complex permanent? Because whether you look at the components of xylem, which includes tracheids, vessels, parenchyma, fibers, or phloem, which includes sieve tube, companion cell, parenchyma, and fibers, we see that they are not made up of only one single type of cells, but we see that many different types of cells come together to perform a common function, right? So they come together to perform a common function, which is why we call them as complex permanent. So here the stem is wrong. Please don't mind. I'll rewrite it. So the question is asking us why xylem and phloem are called as complex tissues, right? So this is the answer. Xylem and phloem are called as complex tissues because they're made up of multiple cells. And all these different cells come together to work in a single unit. That means different cells are coming together to carry out a common function. So in the case of xylem, it is to help in transport of water and minerals, right? While in the case of phloem, we see that the main function here is to help in transport of food, which is why we see that this right here is the answer. Okay. Who is this? Vamsi Bacha, don't spam in class, please. Nobody is free here to play Free Fire. Okay? Awesome. Ma'am, why xylem and phloem? They asked me in exam. Pragati, this is the reason, right? See, Pragati, why is it called as complex permanent tissue? Because they are made up of different types of cells. Not one type, but different types of cells. Yes? And all these different types of cells come together to do a common function. What is a common function? In the case of xylem, we see that xylem is a type of tissue which helps in transport of water and minerals. While on the other hand, phloem helps in transport of food. Ma'am, how do non-green plants perform photosynthesis? Non-green plants, Tarun, may either, there are 1 and 2 cases. Non-green plants, they may either not do photosynthesis and they may exhibit parasitic mode of nutrition, right? Or maybe some parts of their plants are still green that carry out function. Yes? Okay. How many are remaining? We have a long way to go. Awesome. Now let's move on to the next question, right? Ma'am, this question is, this is the question, why do they contain different cells? Nahin. Question is asking why is it called as a complex tissue? They called complex because they are made up of different cells. And all these different cells, you imagine, they are all carrying out different functions, right? So if you look at xylem, again to understand this, here tracheids and vessels are mainly what is responsible for transporting water and minerals, right? Parenchyma to an extent also helps in transport, but mainly helps in storage of food. Fibers here provide mechanical support. So individually they're doing different things, but they also work together, right? They also work together to carry out a common function, that is transport, which is why it is a complex tissue. Ma'am, why? Ma'am, मेरा question है कि why they contain different? Why do they contain different? That is their nature. To be honest, that's the nature, right? Because see, as plants have evolved, right? As plants, see, in the case of plants also, if you see, there is a certain kind of evolution where we see that there are simple types, and as their body started to become complex and complex, they realize that they need efficient transporting systems. So in order to bring in an efficient transporting system, they are made up of those different cells. So I hope that I have answered your question, Pragati. Okay? Now, Tripti, this is what you were asking me, right? This is what you were asking me about squamous cells and everything. Now, I'll explain in detail. So, you have 4 questions and 2 marks, which means you'll get half mark for giving me correct answers here. Lining of blood vessels, they are made up of what? I'll write your answers first. Lining of blood vessels, everybody is telling me, ma'am, it should be simple squamous. Okay. Now, what about lining of small intestine? Yes? Lining of small intestine, what is it made up of? Yes, xylem and phloem together are known as vascular tissue. Very good. Columnar epithelium. Okay. Columnar epithelium. Lining of kidney tubules. What is lining of kidney tubules? Very good. Cuboidal. Last, epithelial cells where cilia is found. Very good. It will be ciliated. So this is your answer. Now let's have a look. Now we have squamous epithelial tissue, right? Now see, squamous epithelial tissue that is there are basically these flattened irregular shaped cells, okay? They are flat cells which have a prominent nucleus, yes? And we see that because they are epithelial cells, they do not have any space between them. Now why? Now squamous epithelial has 2 types. You have simple and you have stratified. Now what is simple? Simple means simple. They have only 1 layer, okay? But it is not complicated, right? It is not complicated. That means there are not multiple layers. But it's a single layer, so we call it as simple squamous. Yes? So we see that here it is called as simple squamous. Where do I find it? I find it in the inner lining of my mouth, in the inner lining of blood vessels, inner lining of alveoli. What is their function? Apart from protection. See, their primary function is protection. But apart from that, in places like alveoli and in the case of thin blood vessels, they help in diffusion also. Now the next question is, what is the lining of small intestine made up of? Small intestine is made up of columnar cells, right? We see that they are made up of columnar cells which are tall. So it's like a pillar, like a column. And we see that the nucleus is found more towards the lower end. Where will I find them? Normally in the inner linings of the intestine. And apart from protection, they help in absorption also, right? So they help in absorption. Then we have cuboidal epithelium. So here it's with respect to its shape. It is cube-like, right? Found in inner linings of kidney and salivary ducts, and they provide mechanical support. No, Pragati, not at all. Please don't feel that way. I mean, I was trying to understand your question. Maybe I also took it wrong now. Please don't feel that way at any point, yes? See, I always tell all of you, please feel free to ask questions. Please feel free. I'll try my best to clarify your doubt. My job is that, no, to make sure that if you sit with me for 1.5 hours, you should not go back thinking, "Arey, I sat in Ashwaraman's class and didn't understand anything." So my job is to make sure that you all understand it. I try my best, right? ठीक है. Ma'am, why are small intestine not made up of glandular epithelium? See, glandular epithelium is a type of columnar epithelium, right? So when columnar epithelium or certain epithelial cells get modified, right? So when they get modified, we see that they can become glandular when they have or when they achieve the ability to secrete substances. So that is when it will become glandular, okay? So we will find glandular cells in the small intestine also. Okay. Ma'am, why are they cuboidal, cube-like and not cuboid-like? That's a good question, but we call it as cuboidal only. That's something I will think about, okay? That's a good question. I'll think about it and come back to you on it. Awesome. Yes, Kamal Preet, ask me your doubts. Anjum, what are you referring to? Please don't get distracted. ठीक है. Now, we know that when certain epithelial cells have cilia in them, we call it as ciliated epithelial cells, which we find in the oviduct, we find it in the trachea, yes? So, we find it here. Awesome. So, this is the answer, and if you give this, you will get 2 marks, okay? Now, very quickly, students, how about we take a 5-minute break? We solved 15 questions. 15 more to go. But before we do that, quick water break. Yes, I'll give you a water break. मैम सीलिया होता है hair like structures, ऐसे ऐसे होता है, and they go back and forth, right? They lash back and forth, so thereby they help in movement. Yes, awesome, no problems. Afia, cilia means hair like structures. So jaldi se students, we will resume at 6:15 p.m. Take a water break, 15 more questions to go. Whatever doubts you are asking, like Rajiv, many of you, I'm going to be having questions based on that. So I will come back at 6:15 and we will quickly have a 5-minute water break, right? 5-minute water break and we will be back. Kamalpreet, I will solve your question. Don't worry. Yes, I will take all your doubts. Please don't feel कि I will not take, but we will be back soon. Okay? Jaldi se 5 minutes, take a water break and we shall be back. Hello students, we are back. Just give me one moment. Okay, so we are back. Gopal Bacha, I couldn't come to the 6 to 8 class because I'm here with your seniors. But Ankita ma'am is there with all of you, right? Yes, I am back. I did not realize that I kept my pen here and I went looking for the pen all over, right? Yes, I will give you strategies for improvement. Oh, I missed the question. I missed the question where I could have given you that, but maybe towards the end. Harini, I've solved 15 questions, 15 more to go, right? We will do, see, this whole month I'll only keep doing these 3 chapters over and over. So you have nothing to worry about, right? So don't worry. Ma'am, how green color is governed? So basically, Tarun, there are specialized cells which produce chlorophyll, right? So if chlorophyll producing cells, if chloroplasts are present, then we know that in such cells, photosynthesis can happen. Now, there are some plants, some algae which appear non-green, but they have chlorophyll or certain type of chlorophyll in them which allows for photosynthesis to happen. So again, it is not always the case, yes? Why are xylem and phloem called as vascular? They are called as vascular because they help with transport. So keeping this in mind, right? So keeping this in mind, let's have a look at question number 16. Yes? Okay. Aditi, watch the video from the beginning in 2x. You will be able to do it. Give 4 points of differences between bone and cartilage. Students, this is a very commonly asked questions in the examination, where they will ask you to differentiate between bone and cartilage. Now, if many of you have already given exams for tissue, I am sure you would have got a very similar question. Somebody at some point has got the question to differentiate between bone and cartilage. Yes or no? I am sure some of you have already got this right. Yes, very good. See, now both bone and cartilage that is there, they are both connective tissue, right? They are both connective tissue. Now, in the case of bones, if you see, we see that they are hard, they are rigid, and they are non-flexible. That means they are not capable of being flexible. While on the other hand, when you look at cartilage, right, we see that they are flexible and they are not very hard, right? So they are, I would say, they are less rigid. Now, here we see that when you look at bones, right, we observe certain pores which are present. So we say that they are porous while cartilage is non-porous. Now, main important thing here is that bones are rich or they are supplied with blood vessels, while cartilage on the other hand lack blood vessels. Now here if you see in the case of bones, and if you look at the matrix of the bones, right, we see that they are made up of calcium, they have depositions of phosphorus that imparts that rigidity. While here the matrix in cartilage is mainly made up of a lot of proteins like your hyaline proteins, yes? While on the other hand, if you see functional wise, what is the function of bones? Their main function is to provide framework to the body, right? They are responsible for giving protection and they also help with locomotion. While on the other hand, if you look at cartilage, we find cartilage located between two bones and they reduce the friction, right? So they are present between two bones and they reduce the friction between them. So that is their function. So based on this, you can give ample amount of differences. But when you are writing the answer, you can differentiate between the two. And you can write about the cells also. Thank you, Siri, for reminding me, right? So you can write about how bones are made up of bone cells called as osteoblasts or osteocytes, while cartilage is made up of cartilage cells which are called as chondrocytes, right? Okay. MCQs are done, lucky tag. You can rewind in the beginning. I've taken MCQs then. Yes? Thank you. Now let's go on to question number 17. Yes, we will do it in this series. Sanvi, thank you for that. Ma'am, okay. Ma'am, gone super. Thank you. Very good, Poshali. Very good. Okay. Abhi, now we'll go on to the next one, which is 2 marks on explaining briefly between protective tissues. Right? So you have to briefly tell me about protective tissues in plants. Let me add that also, right? So what is the main function about protective tissues? So first and foremost, all of you need to tell me, right? All of you need to tell me where are, for Abhimanyu, for plants. So where do we find, or what are the types of protective tissues? Hi Darshan, hello. Where do we find them in the first place? So mainly in plants, our protective tissues include our epidermis, right? So we have it in the leaf, we have it in roots, right? We have cork. Now what is the main function? Yes, protect the body from wear and tear, protect it from pathogens. Exactly. So in the case of leaf epidermis, the primary function is to protect it from any kind of parasite invasion, prevent any mechanical damage from happening, preventing loss of water. While in the case of root epidermis, we see that although their primary function is to do protection, they also help in increasing surface area for water absorption. And cork that is there is something that we observe in older plants, right? Where as the plant grows older, the protective tissue starts to undergo various changes. Now we know that we have a lateral meristem, and in that we have something called as a secondary meristem, which undergoes certain changes and results in the formation of cork cells, right? Now these cork cells are again dead cells that are there. So they are made up of dead cells, and their main function is again to protect it from any kind of mechanical support, right? And mechanical injury. So here if you see, you can make a table, right? You can make a table and you can write it down based on leaf epidermis, root epidermis and cork, and what are its functions. Ma'am, in all these lessons, which is the most important, right? Which comes next? I mean, marks wise, is it tissues on the top? Okay. See, one thing is that marks wise, it's a little difficult to predict what is top, what is first, what is second, what is third, because in 9th grade, up until 9th grade, it's based on teacher to teacher, right? Your teacher is who's going to set the final exam paper. It's not coming from CBSE. So it's a little difficult to predict exactly. But if you were to ask me, most likely how your teacher will grade you is fundamental unit of life, improvement, and then it could be tissues. Or in some cases, see, fundamental to top mein hi hoga. Uske baad you will have tissues and improvement. So that could be one in two ways where they play it out. But again, like I said, it will differ from person to person. Aman Bacha, welcome to the class, right? Welcome to the class, and I welcome you to the channel. We are doing a class on, we are starting a new series called Abhyas, where we do a lot of practice. Yes? Ma'am, in our school, exactly, see, that's what it differs from teacher to teacher, school to school. That is the reason. But if it's CBSE affiliated school, tissues and fundamental unit holds the maximum weightage, because lot of concepts you learn here will move to your 10th standard. That is why. Role of epidermis, Rajiv, helps with mechanical or provides protection. So your leaf epidermis and everything provides protection, parasite invasion, and of course minimizes water loss. While in the case of root epidermis, what do we see? They help in increasing surface area for absorption. Now let's move on to question number 8. Students, this is a 2-mark question, and this is very commonly asked, right? Very, very commonly asked. Differentiate between plant cells and animal cells. This from 8th standard you have this question. From 8th standard we are learning this question. No, if you had cell chapter in 8th grade also, this is a question that we are bound to get, right? So here, ma'am, ye samajh nahi aaya hai. Zaid, you will have to be specific as to what you have not understood. This is very easy. Essay you will get marks, right? So now if you look at plants and animal cells, we know that in the case of plant cells, we know that plant cells have plastids like your chromoplasts, chloroplasts, leucoplasts, and we know that those kind of structures exist. While in the case of cell wall, what do we see? We see that cell wall is an additional structure found in plant cells, which is absent in animal cells. Now along with this, we see that plants tend to have larger vacuoles, right? So plants have larger vacuoles, while animal cells have very small vacuoles, which are very few in number, or they may have no vacuoles, right? So here if you see, along with this, during cell division, we see that there are specialized cell organelles in animals called as centrioles that facilitate cell division process. While here we see that there is no such specific cell organelle, and we observe a cell plate formation during this particular case. So in this case, right, in this case what do we see? These are the broad 2 differences. I've given you 4. You just need to write any 2. Write any 2 between this. Ma'am, what is the surprise? I have a surprise test. No mentee today. Yes, thank you, Pragati, for that. Please help your friends out because some of them may be asking some things that you can clarify when I'm teaching as well. So students, this is a most likely question that you can get in the examination. Please star mark this. Tanmay Bacha, what is it that you want? Please let me know. Mentee might happen mostly in the second week. Next week, mentee will be there. Okay? Then we'll do a full syllabus revision also. Okay. Now going on to question number 19. Now very quickly, how many of us have a problem with mitosis and meiosis? Because this question is based on that. Which type of cell division is required for growth and repair of the body, and which type is involved in formation of gametes, right? So this right here is a question based on cell division. Now what is cell division? Now cell division is a process where if we have a parent cell, we see that whatever cell content is there, right? Whatever is present inside the cell, we see that it will make a copy, right? It will make a copy of itself, and we see that it will give rise to new cells, right? It will give rise to new cells, or we say the cell will divide in order to give rise to a daughter cell. Now what contributes for growth and development? We see that it is mitosis. Now you see in our body, right, especially if you take our example, we see that inside the nucleus, we have something called as chromosomes, right? So we have something called as chromosomes. Now these chromosomes that are there are basically rod-shaped structures and they contain the DNA. Now our body has 46 chromosomes. Yes. So our body has 46 chromosomes. Now from here if you see, during mitosis, what happens is that this is a type of division where all this 46 chromosomes is copied on to the daughter cells also. So the number of chromosomes are maintained, right? So we call this as a equational division, where the parent cell will undergo changes and it will divide in such a way that it has copied the information onto the daughter cells. While in the case of meiosis, it is something that we see in gametes, right, or in the reproductive cells, where if the parent cell has 46 number of chromosomes, the offspring will have only 23. Why? Because in reproduction, we see that there is fusion of these gametes. So the number has to be half, right? Only then we see that the overall number gets maintained. Hence, what do we see? During meiosis is a special type of cell division seen in gametes. So this is why we see that mitosis is a type of cell division which is necessary for growth and repair, while meiosis is necessary for gamete formation. So I hope you guys are clear with this concept. Now quickly moving on to question number 20, right? So we will move on to question number 20, where what would happen if the plasma membrane ruptures or breaks down? What would happen if the plasma membrane ruptures or breaks down? Can you tell me? Students, for reference books, first and foremost, focus on NCERT. Uske baad you do NCERT exemplar questions. Then after that, if you want, you can buy a reference book. Now, ideally, if you look at reference books, there are many in the market. But normally, to be honest, if you want to practice, one, if you download the BYJU'S app also, you will get all the collated questions, one. Two, if you specifically want to buy books, I would recommend, let's say for grade 9th, more than Srijan and everything, personally, this is my recommendation. I have seen ki all-in-one tends to have some good questions. So you can have a look at all-in-one, or you can also have a look at some Oswald questions also. Okay? PDF in the Telegram channel. See, look at this particular diagram. Look at the function of plasma membrane. Now, plasma membrane is a membrane that is responsible for separating all the inner, content of the cell, right? Basically, whatever is there, it encloses all the cellular content. Or, ye cell hai, uske andar jo bhi hai, that is protected by the cell membrane. And that is what separates the inside and it is what separates the inside and the outside of the cell. Now, you imagine, yes, plasma membrane gets disrupted. What will happen? Whatever is there inside the cell will come out, right? It's kind of like popping the balloon, right? So we see that it's like popping a balloon. But if you pop the balloon, whatever is there inside the balloon, imagine you fill it with water or you fill it with air, everything will come out, right? So when the plasma membrane ruptures, what would happen? We see that in this particular case, we see that whatever plasma membrane is responsible for enclosing the cellular content, once it ruptures, all the contents that are there are exposed out, right? Thereby it will cause cell death. Yes? So this right here is the answer. Now let's go on to question number 21, everybody, which is going to be match the following, right? So now we will move on to match the following questions. Okay. Ma'am, what is meant by pathogen? Pathogen is basically a substance, that harmful microorganism that causes disease, right? Abhimanyu, all cell will come out of the cell, correct. Okay. Ma'am, Ritika, awesome. Happy to hear that you aced it. Tell me how much marks you get as well. Ma'am, please solve sample paper. Done. We will do it. Very good, Syed. Vascular bundles are basically your xylem phloem together. They form the vascular bundle, right? Okay. So let's have a look at this particular question. This is easy. Arey, thoda late ho gaya. Last 10 questions I'm at. Fluid connective tissue here. What is fluid connective tissue? Fluid connective tissue, I think I have it in the next one. So our fluid connective tissue is basically going to be matched to blood, right? What fills up the space inside our organs? What fills up the space? It is filled up by the areolar tissue, which acts as a packaging tissue, right? It acts as a packaging tissue. Now, what are striated muscles? Striated muscles that are there are those muscles which help which have alternate dark and light bands, right? Which are mainly found in skeletal muscles. So that is another name. What is the function of the adipose tissue? It is to store fat, as we have discussed. Surface of joints, right? If you see, as a joint pay, I told you at the surface we have cartilage that helps in minimizing the friction. Yes? Then what do we have? Stratified squamous, which is mainly found in the skin. So when you are writing the answer, you can write it in this manner and you will get 3 marks for the same. Yes? Aman, I hear that you are not comfortable. It's not comfortable for clearing my doubt. Please help me. Whatever is your doubt, let me know. Right? Please let me know. Very proud, Ritika. No problem. Next exam, you will ace it. Okay? Okay. Very good, Amrita. Very good. Harini, end of the class, I'll explain it once again if time is left, if you don't mind, because at 7, I think there is another teacher who has class. Ma'am, simple squamous nahi hone chahiye. Kaun sa? Ye to match the following hai na, so that's why. Session end at 7 o'clock. Avanika, little bit you are late. Okay, question number 22. Ma'am, but we have to match the pairs in exam. Yeah, you have to match. You have to write the answer like this. See, you have to write the answer in this manner. That is why. Okay? Question number 22. What is a layer and what is a broiler? What are the differences between the two? So you will get 1 mark for telling me what is a layer and what is a broiler, and 2 marks for telling me one difference between this, right? Now, many of you have asked me about the chapter improvement. How do we study? Now, see, improvement in food resources is a very long chapter, right? It is a very long chapter. So firstly, when you are making notes and everything, when you are making the notes for improvement, firstly use a lot of tabular columns to concise the information, right? Next, the initial part of which is the crop part, is super important. So for tabular column may also have the examples. So what kind of strategies, what are the advantages, disadvantages, examples. Next up, this requires you to draw mind maps regularly, right? Mind map is your strategy to go, right? Mind map is your strategy to go. So you will have to make mind maps and do a lot of active recall. Because see, it is little bit memory based. Nothing else, right? It is memory based. So that is why Abhimanyu and many others. So those of you who have not watched improvement, firstly, I request that all of you, sort of have a look at your... look at the one-shot video that I've gone to do it in detail. I've done it in detail for the concept bit of it. Later on for mind maps and everything, we will sort of have a look at it. Okay. Thank you so much. Geeta, what do you mean? Improvement ka PDF I'll send it again if it is not there. Okay? See, what are layers? Layers are basically kind of poultry birds from which, or we rear them to get eggs from them, right? While broilers on the other hand are reared for their meat. Now, if I am rearing an animal for its meat or for the eggs, the nutritional requirements are different, right? So what do we see? We see that in this particular case, in the case of layers, we see that they require more vitamin A, vitamin K in their diet, right? So, broilers that are there require a more vitamin-rich diet for their development, while broilers on the other hand, right? I think it's gotten a little different. Okay. 1 minute. Yeah. This is for layers, right? See, broilers that are there require vitamin A, vitamin K for their supply, for their diet, and they require a high amount of protein and fat as well in their diet, right? And we see that they are reared for their meat. While on the other hand, if you look at the nutritional requirement in layers, it is slightly different, right? So theirs is slightly different when compared to your broilers. And unlike broilers, they don't need too much of protein and fat, but rather they would require something very different in order to give high quality eggs, right? Which is why the nutritional requirement in both these cases are different. So when you can write this answer in a clean manner so that you will be able to give the final answer. Yes? Ma'am, foreign broiler gives more meat. See, foreign varieties that are there in some cases or exotic varieties are reared both for the meat as well as the eggs, right? Okay. Now let's move on to the next one. Now Rajiv and many others who had doubts in ma'am types of muscle and their location. Yes? Hello bacha, hello. Please don't spam bacha, please don't spam, but hello to you. Difference between pathogen and pest. Pest is an unwanted insect larvae. Pathogen is a disease causing microorganism. Yes? Okay. So let's have a look at this particular question. Here we have to talk about the 3 types of muscles and their location. So now what are the 3 types of muscles? Now the 3 types of muscles that we have include, or the 3 types of muscular tissue include striated muscles. It includes unstriated muscles, and we have cardiac muscles, right? Now in this particular case, if you look at striated muscles, they are also known as skeletal muscles, or they are also known as voluntary muscles, right? So we can also call them as voluntary muscles. Now if you look at its structure, now they are skeletal muscles. No, that means they'll be attached to the bones, right? So if this is a bone, these muscles will be attached to it, like your biceps and triceps. They're all examples of skeletal muscles. Now if you look at it structurally, right? If you look at it structurally, we say that they are made up of long, elongated, they are made up of cylindrical cells. And we see that they have these alternating dark and light bands, okay? So they have these alternating dark and light bands. Now along with this, what do we see? We see that they are multinucleated. They are many in number and they are unbranched, right? So they are unbranched. So what are the keywords? They are unbranched, they are multinucleated, and what are they? They have striations in them. So I can say that they are elongated, right? So we see that they are elongated. Now on the other hand, right? On the other hand, this is the structure. So this is the structure. Now you need to know where we find them. Where do I find them? What is the location? Easy. They are mainly found in the arms and legs, right? So wherever we see, in the arms and legs, we will find them. And what is the function? We can control them. So they carry out voluntary actions. What are my voluntary actions? Walking, running, writing, right? All this is important. So this is the basic structure and this is the function. Geeta, we are going to be doing this for the premium users as well. It's already available and we have a lot more interesting things coming up for you guys. So don't worry, okay? Don't worry. Lot of cool things coming your way. Yes? Awesome. Now next up, what do we see? We have unstriated muscles. Now unstriated muscles that are there are also called as smooth muscles, or they are called as involuntary muscles. Now if you look at its shape, right, I'm going to use green color. We see that they have a spindle shape, right? What do they have? We see that they have a spindle shape. That means it is broad in the center, narrow at the sides. And how many nucleus do they have? They are having only one nucleus. So we call it as uninucleate. And we see that again they are unbranched, right? So no branching is there. Why are they called as visceral muscles? Abhimanyu, they are called as visceral muscles because they make up a lot of the internal organs, right? So we see that they don't have any striation, so smooth. Yes? Now we see that various involuntary actions, right? Various involuntary actions are carried out by them. So we see that mainly where do we find them? I will find them. They make up my esophagus. They make up the intestines, right? So all these I think my spellings are going wrong here and there. Intestines, so on and so forth, right? So that is why we call them as visceral muscles also. Last way we have cardiac. Now your cardiac muscle is little similar to your striated, but they are different. Now cardiac muscles on the other hand, right? We see that these are muscles which show striations. That means those alternate dark and light bands are there. But we see that these guys are uninucleate. So we see that striations are there. Yes, but they are uninucleate, unlike your skeletal muscles. And what do we see? These guys are branched. So branching is there. Yes, branching is available. And of course, we see that they have some specialized structures, right? So they have some specialized structures called as intercalated discs. And these intercalated discs are what help in the rhythmic contraction and relaxation. Where do we find the cardiac muscles? In the heart. What do they help us do? They help in the contraction and relaxation of the heart. So everybody take a quick screenshot if need be. Yes, take a quick screenshot. We will be starting live quiz soon. Don't worry. Rajiv, yes. See Rajiv, Aman, Rajiv is also telling you, please tell me whatever is your doubt. I'll take it. Don't spam. Okay. Yes, Rupinder, I remember you. Hope your exams are going well. Awesome. Ma'am, striated muscles are kind of tendon. No. Tendons are connective tissue. They are different. But tendons will connect your striated muscles to your bones. Yes? Ma'am, BYJU'S is not opening. Go to Play Store and try downloading. You must be able to download. Ma'am, RER and SER are only there to help make plasma membrane or other membranes also. Tarun, RER and SER play a very important role in making the membrane. But RER also makes proteins for other functions. Right? Okay. Awesome. So if you've taken a screenshot, this is how you can give the answer. You can either draw the tabular column that I showed you, or you can do this. Yes? Now let's move on to question number 24. Now this is also a very heavy question that is there, which is differentiate between hypo, iso and hypertonic solution. Now many of you have this doubt, right? What is the difference between hypo, iso, hypertonic? Now see, it's actually pretty easy. Now what is a solution? A solution that is there is basically nothing but a solute or a combination of solute and solvent, right? So solute plus solvent is going to give me solution. Now based on how much of solute and how much of solvent I add, I can make it into hypo, hyper, iso. But how will I know whether it is a hypertonic or hypotonic? So here we need to compare, right? We need to compare it with something. So there is more solute with respect to something. So it is kind of like telling, Aishwarya ma'am got 18 out of 25 in her biology exam. Now how do I know if this is a good mark or a bad mark? Or if this is, more or less? But if I compare it with Anand, right? Now if I compare it with Anand who has got 25 out of 25 in biology exam. Anand has got 25 out of 25. So when compared to Anand, my marks are less, right? I have got less marks. So that more or less is with respect to comparison. Although marks wise you should not compare. Be happy with whatever you get. Yes? Aman, please don't get distracted. So based on this, right, based on this, we make a comparison for hypo, hyper, iso. Now imagine I have a cell, right? I have a cell and I put it in a solution. Now this solution is in such a way that the amount of solute in the cell is equal to amount of solute in the solution. So this is my solution, okay? This is my cell. And how much ever solute is there inside the cell is same as the amount of solute in the solution. So here what do we observe? We observe that even the water molecules, right? There is no difference in concentration in water molecules. Amount of water molecules are also the same. So there is no net movement of water molecules. So where there is no net movement of water molecules, we call it as isotonic solution. But if there is a difference in solute concentration, there could be difference in how much water is available. And we know that if there is a difference in water molecule, right? You imagine if there is a difference where somewhere there is more water, somewhere there is less water. What will happen? Water molecules will move from a region of higher concentration to a region of lower concentration, but where? Across a semi-permeable membrane, right? So it will move across a semi-permeable membrane. And what do we call this? What do we call this? We call this process as osmosis, right? So we call this as osmosis. Now, if in the solution, right? If in the solution there is more solute, right? More solute. That means inside my cell there is less solute. So if there is more solute, it means that there is less solvent, right? There is less solvent in my solution. So where will water go, right? Water will go from the cell, it will move outside. So a hypertonic solution is a solution which has higher solute concentration. And as a result, where will the water go? Water will go from inside the cell to outside the cell, right? There is movement of water. While in hypotonic, what will happen? We see that here the solution, my solution that is there, will have less solute. If it is less solute, it means more solvent. And remember, this more or less solute I am doing with comparison to my cell. So it is with respect, this more and less I am doing is with respect to the cell I am putting inside it, right? So based on this, what will happen? Inside my cell, there is less water, while outside there is no more. So we see that water will go from outside to inside. As a result, what will happen to the shape of the cell? Shape of the cell ko kya hoga? Hypertonic mein cell will shrink. Yes? What will happen to hypo? Cell will swell, right? What will happen in iso? Cell will remain the same, right? So based on this, right? Based on this, what can we understand? We see that these are the major differences between hyper, hypo, iso. And if you do this, you will get 3 marks for the same. So are we all clear? Are we clear with this? Yes? Ma'am, can you do this video with physics next? Physics to Saurabh sir will take. I am not your physics expert, right? Okay, literate boy has started to spam, but I have to put you in timeout for some time, so please don't spam. Ma'am, you changed the way you pronounce osmosis, is it? I hope I did not have a heavy accent on that. Are we clear? Yes, okay. Tarun, I have answered your question on this, RER and SER wala question. Please pay attention now, then only you will be able to hear. Okay. Hybridization, I think I have a question on the same, I'll explain then. Now let's have a look at this question, which is differentiate between chromatin, chromatid and chromosome. Confusion of the year, right? Ma'am, what is chromatin? What is chromatid? What is chromosome? Are we all confused? All sound the same to me, right? Okay, let's have a look at this. Hello Rupa, hello. Yes. Now what are chromosomes? See, I'll do a very simple diagram. I have a complicated diagram with me, but I'm going to make it easy, okay? Chromosomes that are there are basically rod-shaped structures, right? So we see that they are rod-shaped, highly condensed form of the genetic material. Now, in our body, what happens? In our body, we see that the genetic material that is there is DNA, right? And naturally, if you look at it, DNA that is there is basically a double helical structure, right? So we see that this is a double helix or a double helical structure. Now what happens in this case? It is very long. Now it is so long that's almost 1-2 meters long. But this DNA has to fit inside my cell. No, it cannot be sitting to 1-2 meters long. It needs to fit inside. So what do? We start to condense the DNA, right? So we condense it. So it's kind of like if you have a long rope and you need to fit it inside a small purse. What will you do? You will roll it, right? You will roll it and you will shove it inside your bag. So that is, think of it as a way of condensing. So here DNA will condense along with the help of some proteins, and what will it result? It will result in the form, or it will result in the formation of chromatin material, right? What is chromatin? Chromatin is this long thread-like form, okay? So normally we see that DNA will exist as chromatin, right? Chromatin, IN. Those are the thread-like forms. But what happens is during cell division, no? During cell division, this chromatin material that is there needs to get further condensed, right? So we see that it has to get further condensed. So it will form chromosomes. Yes? So it will form chromosomes. Now during this, right, during this cell division process, we also see that there needs to be a copy that needs to get created. So if this is my chromosome, we see that there also needs to be a copy of this chromosome that should get created for cell division to happen. So what will happen? It will make a copy and it will end up getting this X-like structure. So this copy, this identical copy that gets synthesized here, right? This identical copy is what we call as chromatid. So chromosome and chromatid that is there is something that appears during, chromatid is something that appears during our cell division phase, while chromatin is what appears during the normal phase, right? So I hope we are clear with this, students. I hope we are clear. So when you are writing the answer, long thread-like structure is what we call as chromatid. During the process of cell division, it becomes short and thick. So what do we call them? We call them as chromosome. Now this chromosome that is there has all the genetic material. But for cell division, it has to make a copy, right? So that copy that it makes is what we call as chromatid. Yes? Okay, quick question. Ma'am, sister chromatids and simple chromatids, both are the same. No. Sister chromatids are basically the pairs that are formed, right? So a chromatid that is there is on a chromosome, but chromosomes exist as pairs, so we call those as sister chromatids. मैम बायो के एनसीईआरटी सॉल्यूशंस कहाँ मिलेंगे? बायजूस वेबसाइट में मिलेगा या फिर चैनल पे मिलेगा, ठीक है? Axon and lymph. Axon is a part of neuron. Lymph is a type of blood connective tissue that lacks RBCs, mainly made up of WBC and plasma. So keeping that in mind, let's go on to question number 26, right? Question number 26 is describe the composition and functions of blood, right? So what are the composition and function of blood? Now we know that blood is a fluid connective tissue, right? And broadly it has a fluid part and a liquid part. So the fluid part is what we call as the matrix, where we find, I said fluid and liquid, sorry. It has a fluid part and a cellular part, right? So the fluid part that is there is what we call as the matrix, that is the plasma. While the liquid part is basically, the cellular part that is there includes RBC, WBC and platelets. Now RBCs that are there are responsible for transporting oxygen. WBCs help in protecting our body from pathogens and platelets help in blood clotting. So with this, what do we see? You can write it in 4 points, right? You can explain this in 4 points. This is a very common question, right, Rinku? Ye aayega. So you need to write the components. Very, very important. You will get 3 marks for the same. Now quickly we will solve 2 more questions, and question number 30 I will give you as homework. Okay? Now this is a case-based question. Gopal, who was studying in class 9th, thought about what he can do to make his village prosperous. He suggested the village panchayat to integrate farming with animal husbandry. He took the sarpanch to the government agency for loan to buy modern agricultural equipment so that he could buy a safe storage space for cattle. And he also established a biogas plant, right? So how do good animal husbandry? So now what is your question? Your question is, how do good animal husbandry practice help farmers? What is the benefit? 1 mark. Write 2 advantages of having a biogas plant. 2 marks. List any 2 qualities that inspired him. You will get 1 mark for this. So that's how you will get a total of 4 marks here. Now first one is, how do animal husbandry practices help farmers? See, when you have good animal husbandry practices like giving clean environment, giving them proper food, vaccinating them, we see that this right here gives in good yield, right? Good output. So thereby what happens? Thereby maximum output means maximum amount of yield that they get with respect to the product. Whether it be milk, whether it be, meat, whatever it be, they will get good yield. Now along with this, we also see that it also gives them better survival rate so that they can continue to practice it for a long period of time. Now along with this, Gopal has also set up a biogas plant, right? So we see that Gopal has decided to set up a biogas plant. Now what is a biogas plant? We normally use, basically, it's called as gobar gas, right? So you use the cow dung in order to manufacture this. So when he sets it up, raw material is easily available because he set up a cattle farming also. Now when he sets up a biogas plant, he is also using the village to generate renewable energy, right? He is able to generate renewable energy, and this energy can be used for cooking, it can be used for lighting, street lights, and so on. So thereby what happens? It improves the sanitation also of the surrounding. So overall, what do we write? For first one, for 1 mark, how does it benefit the farmer? You can talk about increased output of milk, meat, eggs in case if they start poultry. Proper maintenance gives maximum yield. You can write about other advantages also. The second part of it, what are the advantages of having a biogas plant? Renewable sources of energy, can be used for cooking and all of that. Also helps with sanitation. Then of course, what do we see? We see the last bit of it is, write any 2 qualities that inspired him. What are the qualities? See, he took initiative, right? He took initiative to ways in which he can bring prosperity to the village. And along with that, he was also able to bring in the government panchayat and the village panchayat and the agencies so that they could establish this. Thereby what happens? They are able to grow, right? Okay. So students, will move on to question number 28. I may need 5 more minutes, Manu. So, can we ask, students, 1 minute, can we ask Saurabh to come here? Okay. Oh, fine. 5 more minutes I will need for sure to wind up. So, let's have a look, students. Next question is question number 28. Cells were first discovered by Robert Hooke. So many questions are there. Kamalpreet, I will take your questions. I will take it. But let me finish the existing questions that we have, right? So we see that cells were first discovered by Robert Hooke in 1665. We saw that Leeuwenhoek improved the microscope. Robert Brown did, found the nucleus. Purkinje found this. Now based on this case, you have to give me the answer. Now in case-based questions, half the time the answer is always there in the question only. So who discovered cell? Easy, right? Who discovered the cell? It was Robert Hooke, right? He discovered, it's given in the question. Next up, who discovered the nucleus in the cell? Was it Robert Hooke, Leeuwenhoek, Robert Brown, or Schwann? Who was responsible for discovering nucleus? Now in your question only they have said that Robert Brown was the one who discovered the nucleus. So in this case, what would be the answer? It would be option C, which is Robert Brown. See, case-based 4 marks is something you can easily get, right? You will be able to easily get it because answer is most of the time, the answer is there in the question. Now, who coined the term protoplasm? Bold letters में question में ही है. Purkinje in 1839 coined the term protoplasm for the living part, right? Okay. Who is this Brown Munde and all that? Hey guys, please don't get distracted. Ma'am, you didn't explain diversity pura. I will explain it pura, don't worry. I'll take another class where I'll do full 5 kingdom classification. But that requires me to take a 5-6 hour class. That's how much content that is there in that. So I will do it, but you guys should tell me, ma'am, I will come to class. Not that ma'am will take class and then there are like 10 students in class. No? Very good. So in this case, it is Purkinje. Last bit. What is protoplasm? It is a unit of life. It is a cell organelle. It is the fluid substance or the living components. Yes. And of course, we have cytoplasm as well. So what would be the answer? Easy. It is the fluid substance that is there. Now we will go on to question number 29. So here it is a 5 mark question. which means that 1 mark each you will get, sorry, 1 mark each that you are going to get for giving me the answer. These are give reason questions, right? So here, what is the first part? First part says that we have taken dry apricots, right? So dry shriveled apricots, if I put it in pure water, right? If I put it in pure water and then, so first I put it in pure water and then if I transfer it to a sugar solution, what is going to happen? Yes? Hello Harsh, welcome to the class. Welcome. Okay. Yes, Abhimanyu, today I will send it. I think I sent it to the team, but they have not sent it, so I'll only send it directly. Ma'am, I got 26 question as case based. Is it? See, you have a dry apricot kept in pure water, which means in the first case, it is going to swell, right? The water will enter into the dry apricot and it is going to swell. Now, at the same time, if you put, which means it's come back to normal. Now, if you put a normal cell in sugar solution, what is going to happen? It is going to shrink again. So, it will consecutively shrink. So, this is what is going to happen in the first case. It will swell first and then it will shrink. Now, the next one is what somebody was asking me. If I keep a red blood cell in a concentrated saline solution. So, I have a concentrated saline solution. And now, what am I going to do? I am going to keep a red blood cell. What is going to happen? Yes, very good, Lavi. Exosmosis, right? So the water content that is there will come out. It will lose water immediately and the RBC will shrink. Yes? So this is what is going to happen. Now the next one is if the plasma membrane of the cell breaks down. So here, how to do these questions? You have to do it one after the other. It is give reason. So you will have to write it separately and you will get 1 mark. So if if the plasma membrane of a cell breaks down, what is going to happen? We have already discussed. Cellular content will leak, cell will die. If I take some leaves in boiled water first and then I put a drop of sugar syrup on it, what is going to happen? So if I boil the leaves of a cell, right? If I boil the leaves of a cell, we see that it is going to die. And if I put a drop of sugar water in it, do you think anything is going to happen? See, if I boil the leaf, the cells have died. If the cells have died, will it do exosmosis or endosmosis? Can they do that? No, if the cell has died, what will happen? We see that there will not be basically no intake. So here we'll see that in the dead part of it, the sugar solution will exist, right? So that is one thing. If Golgi apparatus is removed from the cell, so this is the last part. If Golgi apparatus is removed, what is going to happen? We know that a lot of protein modification happens in Golgi. We know that proteins are transported to different parts with the help of Golgi. Now, if there is no Golgi, no lysosomes, no secretions, hence this is the answer. So take a minute to take a screenshot of this part. Yes? Everybody take a screenshot. So this is the first part where you will get 1 mark. What will happen to the red blood cells? This is what will happen. What will happen when plasma membrane is not there? Ho gaya. On boiling, what is going to happen? This is what we discussed. This is very important. Okay? That part is very important. And this is the last bit. So you will get 1 mark for each of it. Yes? Okay. Now the last question that I have, I told you I'll give you homework, but quickly I think I will solve it up. Right? Last question is explain the structure of neuron with the help of a labeled diagram. So we know that neuron that is there is the structural and functional unit of the nervous system. What does the nervous system do? It helps in transmitting signals, right? Information to different parts of our body from the outside and within internally is communicated with the help of neurons, right? So basically neurons is what makes up your nervous system. Now how do they communicate with each other? With the help of electrical impulses, right? So they are communicated with the help of electrical impulses. Now the neuron has a cell body which is also called as cyton, and from the cyton there are these cytoplasmic projections called as dendrites. Now these dendrites are what receive information, okay? Where does information get received? It is by the dendrites. So from the dendrites, it will go to the cell body or the cyton, which has a prominent nucleus. Now from here, we see that there is one long extended projection. That is what we call as the axon, right? The axon that is there is protected by an insulating sheath, which is called as, what do we call it as? We call it as the myelin sheath, right? What is the function? It provides or acts as an insulator. And end may the axon ends as axon terminals, right? So this is where the impulse will reach. From here, the dendrites of the next neuron will take it up. So when you're writing the answer, you can write it in this format and support it with a diagram. So remember students, diagram in this right here is very, very important. Yes? Okay. Difference between semi-permeable, it means that semi-permeable is with respect to size, right? Selectively permeable is what your cell membrane does, which is selective based on functionality, type of molecule, so on and so forth. So students, if you look at it, with this you have solved 30 questions with me. Imagine you've stayed with me till the very end to solve 30 questions. Now, are we feeling more confident, right? Are you feeling more confident about the concepts? Let me tell you all one thing, okay? If you pay attention in class, this is all going to be good for you. If you don't pay attention, this is what you are missing out on. So your homework is going to be a small test. So if you look at the description of this video, there is a Google form where we have given you about 5-6 questions. Now your job is to give the test, right? And once you write the test, let me know in the comments how much you scored, right? Give me your final score in the comments of this video. And those students who get full marks in the first 1 hour after this video has been streamed, which means by 8 o'clock, all of those entries that have scored complete full out of full marks, we will feature you in the community post, and we will make this a regular activity. So everybody jaldi say go and give the test and see how much you've understood and if you have aced it, right? Now I know many more doubts are pending from your end, so I request that you let me know doubts in the comments of this video. And if you enjoyed this class, right? If you love the Abhyas series, if you love how much we are putting in effort for you, and if you feel this is the best platform for exam prep, do not forget to hit the subscribe button on the channel. And in case if you need more and more, right? If you need more practice, more concept bites, or maybe something like that, let us know in the comments. We will definitely take it up. So thank you so much everybody for staying with me till the very end. I will see you all soon. But up until then, take care. Lots of love to all of you and bye-bye.